This video will be going over the reading from the genetics chapter starting on page 30 from our review book. I recommend, as you're watching it, to stop and do any of the highlighting or writing, annotating that I'm doing. I'm going to move through it pretty quickly to keep this video nice and short. Living organisms have a set of genetic instructions that determine the characteristics of their structures and functions. The genetic instructions are passed from parent to offspring through a process of reproduction. During inheritance of these traits, the genetic instructions can be changed, leading to variation. Genetic instructions are found in the form of a code within DNA molecules. Each set of genetic codes, or DNA, is unique to each individual organism. Humans have used techniques to alter genetic information. Through breeding practices and biotechnology, humans are developing new combinations of genes and new varieties of organisms. Organizational relationships. Starting with the largest on the left, we have the cell, which contains the nucleus. Within the nucleus, we find chromosomes. Chromosomes are molecules that hold the DNA. Segments of DNA are genes, and then the smallest are the molecular bases. Now, moving on to DNA's role. In all living organisms, cells stored coded genetic information in the form of DNA. DNA is composed of nucleotides, which consist of sugar, phosphate, and molecular bases that form genetic sequences. These molecular bases are represented by the letters that are the beginning of their molecular names. A is for adenine, T for thymine, C for cytosine, and G for guanine. Molecular bases form complementary pairs. A pairs with T and C pairs with G. These bases form sequences within DNA called genes, which code for specific proteins that determine an organism's traits or characteristics. Genetic material is stored in the cell for efficient replication practices as well as protein synthesis. DNA is coiled and packed into structures known as chromosomes. Chromosomes are located within a nucleus or genetic area within a cell. DNA's double helix structure allows it to serve as a template for DNA replication. DNA unzips using enzymes, and new nucleotides attach to exposed strands, forming two new identical DNA strands. This process allows for continuity of genetic material to be passed from parent to offspring. This process is an essential part of asexual as well as sexual reproduction. Asexual reproduction results in offspring that are identical to the parent, while sexual reproduction results in offspring that resemble but are not identical to the parent. The processes of mitosis and meiosis rely on the replication of DNA. Mitosis is the process of cell division that produces identical daughter cells for growth and repair, whereas meiosis is the process of cell division that produces sex cells or gametes. Each process uses DNA replication. DNA also serves to store the codes for production of proteins, which are vital to the proper functioning of cells and all living things. Within the cell, protein synthesis occurs when coded genetic information is copied and transferred from the nucleus to the ribosomes. RNA's role. The copy and transfer of genetic information involves a second nucleic acid, RNA. RNA is single-stranded and uses a molecular base represented by U instead of T, making the complementary pairs A with U and C with G. In the ribosome, amino acids are assembled into chains, forming a protein molecule. The sequence of amino acids is determined by the sequence of molecular bases on the copied RNA strand. A sequence of 3 bases on RNA, known as a codon, codes for a particular amino acid. A universal chart allows geneticists and researchers to convert RNA code into an amino acid. The original code for every protein begins with DNA. Each protein has a specific shape that determines its function, all based on the sequence of those amino acids. Expression of genes. When gene sequences in DNA are accessed to make specific proteins, those genes are said to be expressed. The phrase, genes are turned on, may also be used to describe gene expression. Gene expression is regulated or controlled by several factors. There are internal controls that allow genes to be expressed when proteins are needed. The environment can also influence gene expression. Factors such as sunlight and temperature can determine whether genes may be expressed. Mutations. Changes in genetic sequences are known as mutations. A gene sequence can be changed through deletion, where a portion of the genetic code is lost or missing, through addition, where a section of genetic code has been added to the existing sequence, or through substitution, a process where information from one chromosome is traded with another chromosome. During meiosis, closely aligned chromosomes may trade sections in a process known as crossing over. This results in a new genetic makeup in the chromosome. Some mutations may promote genetic variation, becoming either beneficial or detrimental to a population. There are many mutagenic agents that can cause mutations within DNA, including chemicals, UV rays, X-rays, and other types of radiation exposure. In order for a mutation to be passed from one generation to another, the DNA in a sex cell, egg or sperm, must be changed. Mutation to DNA in body cells will not result in a mutation being passed to the next generation. Gene manipulation. Humans have altered genetic information through selective breeding to create enhanced varieties of plants and animals. By choosing organisms with the most desired traits and breeding them, farmers and breeders have created many new varieties. With greater knowledge of DNA and genes, humans can now use genetic engineering to manipulate DNA to produce new traits within existing organisms. By using restriction enzymes that cut DNA, scientists can cut, copy, and move DNA segments from one individual organism to another. When the DNA segment is inserted into the DNA of another organism, such as a bacteria, the altered DNA will then contain a foreign DNA segment and also express it. For example, the human hormone insulin is now genetically engineered using bacteria, see diagram 5. Researchers have also been able to clone organisms by inserting a whole set of genetic instructions for an organism into an egg cell. Cloned organisms will contain genetic information identical to the donor parent organism. Using these techniques and their increasing knowledge of genetics, researchers have been able to locate disease-causing genes and develop preventative measures to help fight those diseases. Researchers have also genetically engineered hormones and enzymes that could provide economical advantages and produce fewer side effects when used in medicines. Other genetic techniques, such as gel electrophoresis, see diagram 6, are used to identify individuals as well as determine paternity, based on the genetic information available. The use of genetic engineering so far has led to advances in agriculture and medicine that have benefited mankind. Additional information. Many genetic disorders are a result of a change in the genetic sequence of a gene. This change may lead to a disruption in the synthesis of necessary proteins. Several genetic disorders include cystic fibrosis, hemophilia, and sickle cell anemia. Identical twins are genetically identical, having the same DNA, and are always the same sex. But different environmental influences throughout their lives affect which genes are switched on or off. Thus, the twins will show different characteristics based on that environmental effect on gene expression. RNA has several forms. mRNA is utilized to transcribe and copy the genetic code and bring it to the ribosome. tRNA brings amino acids to the ribosome to be assembled into proteins based on the genetic code. Sometimes chromosomes fail to separate during the process of meiosis, resulting in gametes that have either an additional chromosome or a missing chromosome. This is known as non-disjunction. When these gametes are joined during fertilization, the resulting offspring will have one extra or one less chromosome from the normal species chromosome number. A person who has an extra chromosome, number 21, has Down syndrome, a disorder that exhibits some health abnormalities. Many environmental disruptions that have been caused by man can lead to genetic mutation. This is especially true in places that have had serious nuclear disasters, like the Chernobyl power plant, located in Russia, and the Fukushima Daiichi nuclear power plant, located in Japan. Because DNA is unique to each individual, it has become useful in many biotechnological procedures as well as in forensic investigation. Scientists have successfully mapped the human genetic code. This mapping is called the Human Genome Project and can provide valuable information to researchers and geneticists.